Hey guys, membranes in a nutshell. Membrane proteins especially. Of course, the membrane's a phospholipid bilayer, and we have a variety of proteins embedded in it. Remember, it's that fluid mosaic. It can move around, but it's made up of a lot of small different components. So that's our fluid mosaic model. Besides the phospholipids, those small components are proteins of various kinds, and we use them in various ways. But first, we'll talk about channels and pumps. So here's my membrane. I've got, let's say this is a channel and this is a pump, and this is the X pump. It handles ion X. And this is the X channel. It handles the X ion also. So let's say I've got a whole bunch of X out here, very high concentration. High concentration of X. Remember, the brackets just means concentration. And then here I have very little X. So this is a low concentration of X. So X has to be something that's hydrophilic because it doesn't want to be in this hydrophobic barrier in here. Remember, all those tails from those phospholipids are in here, and that's very hydrophobic, and it prevents anything hydrophilic from passing through the membrane without help. So a channel allows these guys to move according to their concentration gradient, going downhill, going from a high concentration to a low concentration. That's pretty straightforward. A pump is how you move things against their concentration gradient, and that requires ATP. And that's what makes it a pump. So that's pretty simple. Remember, we have combinations of these things that we can use. We can use co-transport and things like that to do secondary transport. And if you don't really remember that, it's worth going back to your cellular toolbox. I've got it all described in there in a nice little PowerPoint. So you can go back to that PowerPoint from first semester. Cellular toolbox is one of the things you should definitely be looking at anyway. What else happens in membranes? We also do signal transduction. So here's my membrane. Here's a membrane protein, and I'm going to draw it, I don't know, just like this. There we go. It's a big lumpy protein, but it has an active site. It has a place where something can bind. So this is inside the cell, this is outside the cell. And the way signal transduction works is that some outside signal, sometimes called the primary messenger, right? It's usually a hormone, but it can be all kinds of things. It could be a paracrine hormone from a nearby cell. Over here, here's another cell. It may release this guy, or maybe some faraway cell in the body and it travels through the blood and gets here. Or maybe it's even attached to that. So we talked a little bit about cell-to-cell communication. Direct cell-to-cell interaction is where you have a membrane protein binding a membrane protein. And so now both of these cells are both aware of the other one's presence. But whatever that messenger is, whatever it is the thing that tips off that signal, we're going to get this same thing happening. When this binding happens, when the ligand binds the receptor, it causes a shape change. Remember, anytime you bind something to a protein or take something off the protein changes shape. In this case, you get a shape change that makes this inside do something new. And usually it's some kind of enzymatic function. It kicks off some other protein, makes it do its thing. It becomes active. And so it goes and does something and makes some other protein active. And maybe that guy activates, 20 or 30 other proteins that then each goes and activates 20 or 30 other proteins, right? And you get this amplification, right? So our first step is reception. We receive a signal. And then this whole thing is transduction, where the signal comes across the membrane and then begins to cause things to happen. And then you're going to have some cellular response. And it may be that somewhere down this kinase cascade, down this road, there may be a secondary messenger. Sometimes it's cyclic AMP, or maybe it's calcium ions. Something like that's going to happen. We've talked about a lot of different examples of this. But that response is going to come because of that secondary messenger after this signal transduction pathway. So reception, transduction, response. That's the 3 steps to signal transduction. It's basically a way that an entire cell can respond to a single molecule's presence outside the cell. The primary messenger is not brought into the cell. It stays outside. It causes a shape change in the receptor protein, which makes the inside of the cell aware of that and does things because of that. So that's pretty straightforward, and we've covered that a whole bunch of times. Again, that's covered in that cellular toolbox unit. So that's a couple of the things about membranes you guys were asking about. A few other things that kind of go with that. Like I said, secondary transport, membrane potential, right? Having a voltage difference outside and inside of the membrane, right? And that's usually maintained by a pump of some kind of positive ion. For us, it's almost always sodium. We can do lots of different things with these transport proteins. And with signal transduction, we can kick off, cell division because of some signal we received outside the cell. We can secrete some enzyme or some proteins from vesicles because we receive some signals. There's all kinds of things that can happen. Really, the things you need to know are what are the things that a cell can do. So they may give you a question that requires that you understand how the cell does this thing. So that cellular toolbox, that'll come in handy. You definitely want to spend some time with that. That's membrane proteins in a nutshell.